Welcome to Catalyst Medhub, where we ignite understanding and make learning easier. In our today's class, we shall be discussing on urea cycle, which is an important metabolic pathway or an important metabolic process that leads to the conversion of toxic ammonia into non-toxic urea. Ammonia, as we know, is produced during the amino acid metabolism or nitrogen-containing substances. And ammonia have to get rid of the body because it's very toxic to the body, especially to the brain. Ammonia is very toxic to the brain because it decreases the rate of ATP synthesis in the brain. So, introduction. Urea cycle is simply referred to as a process that converts ammonia into urea. The starting point of the urea cycle is the ammonia and the end point is urea. So a process of converting toxic ammonia into the urea is called the urea cycle. It is also called Krebs-Enzyme cycle or ornithine cycle. So that is the introduction to the urea cycle. Next is site or location. Urea cycle chiefly occurs in the liver and it is compartmentalized. That is, some part occurs in the mitochondria, while some part occurs in the cytosol. Urea cycle is chiefly occur in the liver because the enzyme, the last enzyme which is necessary for the synthesis of the urea, is mainly or majorly located in the liver. That is the arginase. The enzyme arginase is mainly located in the liver. Though other tissues can undergo urea cycle, but they will not give urea as the end product. For example, the kidney can undergo urea cycle up to the fourth step, but due to the lack of that enzyme arginase, urea cannot be formed or it cannot be synthesized. So the location of the urea cycle is in the liver. And we said it is compartmentalized. It partly occurs in mitochondria and partly occurs in cytosol. That is about the location of the urea cycle. Next we shall be discussing on is the steps of the urea cycle. It is a 5-cyclic step in which the first 2 steps occurs in the mitochondria and the last 3 steps occur in the cytosol. And the steps are, each of the steps have a specific enzyme that it requires. So the steps of the urea cycle are, the first step is synthesis of carbamyl phosphate. Synthesis of carbamyl phosphate. The second step is formation of citrulline. Formation of citrulline. The third step is synthesis of arginosuccinate. Fourth step is cleavage of arginosuccinate. And the last step is formation of urea. So these are the 5 steps of the urea cycle. Synthesis of carbamyl phosphate and formation of citrulline, both of them occurs in the mitochondria because the enzyme that is necessary for the synthesis of carbamyl phosphate and formation of citrulline, they are located in the mitochondria of the cell. Whereby the synthesis of arginosuccinate, cleavage of arginosuccinate and the formation of the urea, they occur in the cytosol because the enzyme that is necessary for them, they are located in the cytosol. So now, we said the first step here is the synthesis of carbamyl phosphate. The synthesis of the carbamyl phosphate is the first step of the urea cycle, and it is formed by the reaction between 1 molecule of ammonia and 1 molecule of carbon dioxide. We said 1 molecule of ammonia plus molecule of carbon dioxide, they will react together to form carbamyl phosphate. So ammonia plus carbon dioxide will give us carbamyl phosphate. Now, this step is catalyzed by an enzyme called carbamyl phosphate synthase 1. The enzyme that is required in the first step is carbamyl phosphate synthase 1. carbomoyl phosphate synthase 1. Synthase 1. This enzyme is one that catalyzes the reaction between the ammonia and carbon dioxide to form carbomoyl phosphate. And the carbomoyl phosphate synthase 1 is allosterically activated by another substance called N-acetyl glutamate. And the first step here requires 2 molecules of ATP. The first step here requires 2 molecules of ATP. So from here, 2 molecules of ATP will be converted to 2 molecules of ADP. Now we say that the carbomoyl phosphate synthase 1 requires an activator which is called N-acetyl glutamate, that is NAG. Is an activator of carbomoyl phosphate synthase 1. N-acetyl glutamate is formed between glutamate and acetyl-CoA in the presence of an enzyme N-acetyl glutamate synthase, and it is degraded by an enzyme called hydrolase. There are 2 types of carbomoyl phosphate synthase. There is carbomoyl phosphate synthase 1, the one that catalyzes the reaction between ammonia and carbon dioxide in the Urea cycle, and there is carbomoyl phosphate synthase 2. That one is concerned or catalyzes the pyridine nucleotide synthesis. So that is the first step of the urea cycle. The second step is formation of citrulline. Now, the carbomoyl phosphate will react with ornithine to form citrulline. So, carbomoyl phosphate plus ornithine, it will form citrulline. We react with ornithine to form citrulline. So that is the second step. Formation of citrulline is the reaction between carbomoyl phosphate and ornithine to form citrulline, and it is catalyzed by an enzyme called ornithine transcarbamylase. The enzyme beware here is ornithine transcarbamylase. That is the second step. I will say that the first step and the second step they occur in the mitochondria because the enzyme carbomoyl phosphate synthase 1 and ornithine transcarbamylase, which are involved in both steps, they are located in the mitochondria. So this is the mitochondria. Now to the third step. The third step is synthesis of adenosine. Citrulline will condense with aspartate to form adenosine. So here, the citrulline will condense with aspartate to form adenosine in the presence of an enzyme called adenosine synthase. So the enzyme adenosine synthase will condense citrulline with aspartate to form adenosine. So here this is our adenosine. Now this step also utilize ATP. So this step ATP is also converted to AMP plus pyrophosphate, which later becomes inorganic phosphate. So, and again, this step again introduces another amino group to urea, because urea has 2 amino groups, one from the ammonia ion and the other one from the aspartate. The general structure of ammonia is the first amino group is from the ammonia and the second amino group is from the aspartate. During the condensation of aspartate to citrulline to form adenosine, another amino group is introduced to the urea. Now, the next step is the cleavage of adenosine. An enzyme called adenosine cleaved adenosine to form arginine and fumarate, whereby the fumarate will enter the TCA cycle. This is one of the link between the TCA cycle and the urea cycle. the fumarate that is formed due to the cleavage of adenosine will enter the TCA cycle. Fumarate will be converted to the malate. Malate will be converted to oxalate, and the oxalate will undergo transamination to form aspartate again, which will enter the urea cycle. So urea cycle and the TCA cycle, they are interlinked or they are interrelated. So we said the arginine succinate will be cleaved to form fumarate. Here is the fumarate which will enter the TCA cycle and the arginine. So the enzymes that cleave adenosine to give fumarate and arginine is succinase. Now, the last step is the formation of urea. Arginine will be cleaved to form urea and ornithine. So here, the arginine will be cleaved in the presence of an enzyme called arginase. So the arginase will cleave arginine to form urea and ornithine. Ornithine will now enter the mitochondria again, where it will combine with carbamide phosphate to form citrulline. So ornithine in this case usually serves as a catalyst because it will enter the reaction and come out like that. Now, arginine will be cleaved to form urea and ornithine in the presence of arginase, and the ornithine will enter mitochondria through ornithine transporter, where it will bind with carbamide phosphate to form citrulline, and the cycle continues. The first two steps occur in the mitochondria, and the last three steps occur in the cytosol. So this is the mitochondria and the cytosol. This is the summary of the steps that are involved in urea cycle. Next thing is the energetics of urea cycle. The energetics simply means the energies that are produced or hit during urea cycle. First, we should know that urea cycle utilizes 4 ATP. It utilizes 4 ATP. 2 is utilized from the first step, and 2 is utilized on the third step. The enzyme carbamide phosphate synthase utilizes 2 ATP, which will lead to the formation of carbamide phosphate, and the enzyme adenosine utilizes 2 ATP during the synthesis of adenosine. So urea cycle utilizes 4 molecules of ATP. And again, the urea cycle yields 3 molecules of ATP because the fumarate, which will enter the Krebs cycle, will yield 3 molecules of ATP. So the net energy of the urea cycle will now be the 4 that it utilized minus the 3 that it consumed, which will now give us a net of 1 ATP. So the urea cycle requires 1 ATP. That is the energetics of the urea cycle. Next thing we shall be looking at under the urea cycle is the relationship between urea cycle and the Krebs cycle. The urea cycle and the Krebs cycle, they are interrelated together because they are interrelated together majorly by 3 means. First is the fumarate, which is released due to the cleavage of adenosine. The fumarate will enter the Krebs cycle. That is the first link between the urea and the Krebs cycle. Second one is ATP that are generated from the Krebs cycle will be utilized by the urea cycle. The third one is the carbon dioxide that are released from the Krebs cycle will be utilized in the urea cycle. So the relationship majorly occur due to the 3 things. First is the fumarate. Another one again is the ATP, and the last one is carbon dioxide. Now, coming to this diagram, this urea cycle and Krebs cycle. Now, from here, we say the urea cycle, ammonia ion plus carbon dioxide, it will give us carbamide phosphate. The carbamide phosphate combined with ornithine to give us citrulline. And the citrulline will condense with the aspartate, which is coming from the urea cycle, aspartate. that is coming from the urea cycle to form adenylsuccinate. Now, adenylsuccinate in the presence of an enzyme, adenylsuccinase, will now cleave the adenylsuccinate to form fumarate. Now, the fumarate formed from the urea cycle will now enter the Krebs cycle. This is the fumarate entering the Krebs cycle and becomes malate. The malate will become oxaloacetate. The oxaloacetate will now undergo transamination to give aspartate. Now, this aspartate will now enter the urea cycle and combine with citrulline to form adenylsuccinate. So this is one of the link between the urea cycle and the Krebs cycle. Another one again is the byproduct of urea cycle, which is carbon dioxide, will be used in urea cycle. The carbon dioxide that are released will be used in urea cycle from the first step because the ammonium ion will combine with the carbon dioxide coming from the Krebs cycle to form carbamide phosphate. That is another link between the urea cycle and the Krebs cycle. The last one is about their ATP. Krebs cycle give approximately 12 ATP and the urea cycle utilizes 4 ATP. So this is the interrelationship between the urea cycle and the Krebs cycle. Adenylsuccinate will be cleaved to form fumarate and arginine. Fumarate will enter the Krebs cycle, become malate. From malate to oxaloacetate, which will undergo transamination to give aspartate. The aspartate will enter the urea cycle again, combine with citrulline to form adenylsuccinate. Again, the carbon dioxide that is released from the Krebs cycle will combine with the ammonium in the presence of carbamide phosphate synthase 1 to give carbamide phosphate. And the 12 ATP from the Krebs cycle, urea cycle utilizes 4 of the ATPs that are generated from the Krebs cycle. 2 from the first step, which is utilized by the carbamide phosphate synthase 1, and the 2 are from the third step utilized by the adenylsuccinate synthase. So that is the relationship between the urea cycle and the Krebs cycle. The next thing we shall be looking on is regulation of urea cycle. Urea cycle is regulated by 1. There is allosteric regulation. 2. Level of substrate. The chief enzyme that regulates the urea cycle is carbamide phosphate synthase 1. And this carbamide phosphate synthase 1 requires an activator which is called now NAG, N-acetyl glutamate. Carbamide phosphate synthase 1 cannot perform its action without NAG, and N-acetyl glutamate is formed by the reaction between glutamate and acetyl-CoA in the presence of an enzyme N-acetyl glutamate synthase. Now, the factors that increase the activity of N-acetyl synthase is the arginine. So, increase in the concentration of the arginine will in turn increase the synthesis of N-acetyl glutamate, and increase in N-acetyl glutamate will activate the enzyme CPS1, which will in turn regulate the urea cycle. Another one again is the level of the substrate. Urea cycle is regulated depending upon the amount of the ammonia as well as the availability of the aspartate. So that is the regulation of urea cycle. The next thing under the urea cycle is the disposal of urea. How is urea disposed from the body? Urea that was synthesized from the liver, they will diffuse through the blood into to the kidney. Then from the kidney, they will not be excreted with the urine. Those small quantity of the urea, they will enter the intestine where they will be acted upon by the bacterial enzyme called urease. And the urease will break down urea into ammonia and carbon dioxide, from which some of the ammonia there will be escaped through feces. So the disposal is through urine, majorly through urine, and few of them they enter the intestine. Few of them enter the intestine. In the intestine, they will be acted upon by an enzyme called urease, and it will break down urea into carbon dioxide and ammonia. The next thing here is disorders of the urea cycle. The disorders of the urea cycle, the majorly one arises if there is a deficiency of a specific enzyme. Each of the specific enzyme here, if there is a deficiency of specific enzyme in each of the steps, it will lead to disorders of the urea cycle. And the disorders of the urea cycle, they include: one, there is hyperammonemia. Two, there is hyperammonemia. Three, citrullinemia. 4, agenosuccinate aciduria, and 5 is hyperagenemia. These are the common disorders of the urea cycle, and each of them result due to the deficiency of a specific enzyme. The first one, hyperammonemia. Ammonemia simply means increase in the level of the ammonia in blood. So there is 1 and hyperammonemia 2. Hyperammonemia type 1 it results from the deficiency of an enzyme carbamide phosphate synthase 1. The deficiency of the enzyme carbamide phosphate synthase 1 will lead to hyperammonemia 1. Now, hyperammonemia 2, it results from the deficiency of an enzyme called ornithine transcarbamylase. So, the deficiency of ornithine transcarbamylase will lead to hyperammonemia 2. Next one is citrullinemia. Citrullinemia simply means presence or increase of citrulline in the blood, and it occurs due to the deficiency of an enzyme agenosuccinate synthase. The next one is agenosuccinate aciduria. Here, it simply means increase in the concentration of agenosuccinate, and it occurs due to the deficiency of an enzyme called agenosuccinase. And the last one here is hyperagenemia, increase in the concentration of arginine in the blood, which occurs due to the deficiency of an enzyme called arginase. So these are the common disorder of the urea cycle and the enzyme and the deficiency of the enzyme that leads to them. The common symptoms of these disorders include: there is usually vomiting, there is lethargy, there is mental retardation, and also ataxia. And some critical condition, it can lead to coma and death. So, and again, the degree of its severity depends on the type or the location in which the deficiency occurs. The first two here, hyperammonemia and hyperammonemia 1 and hyperammonemia 2, they are very dangerous compared to citrullinemia, agenosuccinate aciduria, and hyperagenemia, because in hyperammonemia type 1 and 2, there is increase in the amount of ammonia, which is very toxic to the body, especially the brain. Whereby for the rest, the concentration of the ammonia is less. What are much here is these substances, that is citrulline, agenosuccinate, and arginine, which are less toxic compared to ammonia. The last thing we shall be talking on under the urea cycle is blood urea. The presence of the urea in blood is called uremia. In normal healthy individual, the concentration of the urea in blood is approximately 10 to 40 milligram per dl. So, increase in the concentration of the urea in blood, it usually arises, or the causes of the increase in the concentration of the urea in blood, they are classified into 3. There is pre-renal, 2, there is renal, and 3, there is post-renal. Pre-renal simply means the causes before the kidney. Renal means the kidney causes, and the postrenal means after the kidney. The prerenal is always associated with increase in protein breakdown. If there is an increase in protein breakdown, definitely there will be an increase in blood urea. Then the renal, most of the renal diseases such as chronic nephritis, acute nephritis, or any of the renal diseases can lead to an increase in blood urea. While by the post-renal, any blockade at the lower urinary system can lead to increase in blood urea level, such as the enlargement of the prostate. So this is the summary of the urea cycle. Kindly subscribe, like, comment, and share. Don't forget to turn on your notification bell so that you don't miss any new video.